Là, c'est ton Hello, we speak in English, right? Can you hear us online or not?
Yes, we can hear you.
Very good. So we're gonna start. So I'm gonna just present ourselves. We are two researchers today. Talk to you about emerging biofabrication techniques of cancer research. So I am Guillermet-Guibert. I am a research director at INSERM, working in CRCT. And here, Morgane Delarue, maybe you want to present yourself? Or?
I'm a CNRS researcher at the last CNRS, or the biotechnology lab.
So Morgane and I have developed approaches together. But today I will present more the biology part, and Morgane will show you what he's doing in the physics part. But both are really complementary. And in particular, I will focus my talk about pre-cancer, meaning steps before cancer. So how we can use these biofabrication techniques for the modeling of the initiation steps of cancer. A little bit of introduction on the disease on which we are working. My lab is working on pancreatic cancer. Pancreatic cancer is a dramatic disease. The expectancy of the patients is really poor. If you are diagnosed with pancreatic cancer, you don't have many chances to survive. And it can happen if you are eligible to surgery. It's only 15% of the cases. Fortunately, still a cancer which is quite rare, but the incidence is increasing in the world, in particular in our countries with our lifestyle. There is an increase of this cancer. So it's quite important to understand why the incidence is increasing, why we cannot treat it, and one way to understand that is to study the initiation step of pancreatic cancer, why it is different from other cancers, and what makes it so aggressive. This is a bit of general background. So yesterday you had a presentation and a teaching by Catherine Müller. And from what she told me, she's presenting you the relationship from cancer cell and microenvironment. Is it right? So what she showed is here, okay, the malignant tumor step. She's studying, and she showed you the importance of what we call the microenvironment on the tumor. So in a tumor, you have tumor cells, but you also have immune cells, fibroblasts, endothelium, and this is what is represented here. And this relationship, intrinsic relationship between the tumor cell and the microenvironment doesn't happen like that. There's different steps. So you've learned, I'm sure, during your studies, the different steps of oncogenesis with the mutate genes, like you have a first hit, second hit of oncogenesis. The same, we can also talk about the same with the relationship with the microenvironment. And in the first steps where the mutations are initiated, there's already a dialogue with the microenvironment. And this changed the tissue very gradually. You have in all the cancer, you have steps which are called pre-neoplastic lesions, and they are different depending on the location. And finally, you reach malignant tumor. Okay, so how do we model this? I think you have started to talk about it yesterday, but the question is how we model that as well. Can we use techniques to understand that? And in particular for pancreatic cancer, which is the topic on which we are working, It's a bit more complicated than this simple schematics in 3 steps. First, initiating cells of cancer can be diverse. It can be it's from the one part of the pancreas, which is called the pancreas. But we know now that not all the cells in pancreas are similar. There is also different type of pre-cancer lesions that can evolve, and the microenvironment around them is different. We don't know exactly how it is, but that's images, for example. And then some of them will evolve in cancer, and that's one of the key questions that we need to answer, why some of them evolve or not. So this question applies to pancreatic cancer, but it's also the same in colon cancer. You can be detected a polyp, and it will not evolve in cancer all the time. For colon cancer, it's not so dramatic. The surgeon is going to remove it, but for pancreatic cancer, we cannot remove pancreas if there's only pre-cancer lesions, because this surgery can lead you to die. It's a lethal surgery, and you have a big risk to die with the surgery. You need to balance and to be sure that these are going to evolve. So today, in my presentation, so that's the context, the disease context. I will start by showing how use modern approaches which were developed in the last 10 years to study established cancers, coming from 2D, 3D, and biofabrication techniques. Okay. And then I will explain you the limits of these studies, and in particular for studying pre-cancer lesions. And finally, I will show you the data from the lab, what we are developing to sort out these issues. Okay, so establish. Oh, yeah, the big, sorry, didn't read myself. You need to read heterogeneity. So first thing, we are always present cancer, cancer, solid cancer, whatever, but in reality, there's not one, we know now that in patients, they are very heterogeneous. So to explicit that, you have here, this is a transcriptomic analysis of all these pancreatic cancer patients here. So that's the mutational background. You don't need to know what it is exactly, but it shows you that each column is a patient. You can see the mutations are not similar between them. And the gene expression is very different between them. You can make classification, but they are very different. Okay. So when I was doing my PhD, we were using cell lines derived from patients. We were only looking at the scratch, only a few types, So one of the ideas that emerged is to start investigating this using primary cells directly from patients. Okay? So this is called primary cell culture. To finish on this slide, why it is important is depending on this molecular heterogeneity, the aggressivity, they are all very aggressive, but some of them are hyper aggressive and some of them are less. Okay, so we change the output for the patients. It means that it might also change the way we treat them, even we don't know yet how to treat any of them, but that's the concept. And also behind that, the dialogue with the microenvironment is different. Okay, so heterogeneity, how do we deal with that? Important. Okay. Also, one of the big, if you want to study the dialogue with the tumor cells and the microenvironment, the best way to do it now is to use in vivo model. Okay. And there's 3 types of in vivo models, and so you've learned that. Either you inject cell lines in an ectopic way, meaning not under the skin, not in the real location of the cancer, or in an orthotopic way, meaning in the tissue. Okay. This is done in immunodeficient mice, so you cannot study the immune cell response. Or you use genetically engineered mouse model. So you mimic it by inducing mutation in the mice, and you study how it evolves. Or you develop cell lines directly from these mice and re-inject them autotopically. Okay. So this is very important, in particular for studying metastases. We do not know how to do better than that. But still, we want to reduce the amount of experiments that need to be done in mice. That's why we are also developing alternative approaches. Okay. And how do we do that? So I've talked to you, like this is my PhD 40 years ago. We were doing only 2D cell lines that derived from patients that we bought in a repository. We had a list of cell lines and we would buy that. Next step is understanding the importance of 3D culture. Okay, so we realized that growing the cell in 3D could better mimic the cell-to-cell interaction. And I'm sure Morgan will specify more behind the physical development of this. So there are different ways to develop 3D culture. Simple ways are to grow the cell in inert wells, where the cell cannot attach to the plastic, so they attach to each other and they make a spheroid. Or the same way, you put them in a drop that you put the other way, and with gravity, they stick to each other and they go in the spheroid. Okay, and then we remove it and you put it with more medium. Okay, so this is really good. You have 3D, but you don't have the heterogeneity that I talked to you before. And the innovation that arrived 10 years ago was what we called organoids. I'm sure you've heard about organoids. What does it mean for you? Like a reproduction of small organ, that the brain, for example. Yes. Yes. So how started not for cancer, from stem cells, from stem cell biology. Exactly. We take stem cell and they reproduce spontaneously the same organization and the same distribution, progenitors and lineage differentiation. And this was started in colon, because in colon you are able to reproduce all the different cell types that arise from the stem cell. So in colon it's quite easy because you have stem cell and they reproduce the epithelium very quickly. So let's say for pancreas, for example, there's no stem cell. Okay, in adult pancreas, there's no stem cell. We cannot do that. So in a similar type of things, people have been taking samples from cancers and derive what we call patient-derived organoids, PDO. That's what you see in papers. Okay, when this acronym is written, PDO, it means that the cells were taken from a patient and they never saw the plastic. They were grown directly in a medium with matrigel or matrix. And they are passaged like that in 3D and freeze down if you want to store them in 3D. Okay. So that's what is explained here. And if the tumor cells are epithelial, they can also reproduce constantly this epithelium, but it will be inverted, right? So the lumen will be outside instead of being inside. So that's how it happens. You take directly from the patient tumors or from biological fluids, because you can also do organoid from circulating cell. It's more complicated, but we can do it now. We embed them in matrix, and we have a specific medium. There's always a validation steps. So you want to verify that what you've taken is really tumor cell. This is done by pathologists. And once you have done that, you dump them in vials. So I think 10 years ago, there was a big worldwide effort to bank all these patient-derived organoids all around the world, and in France as well. So now you have banks of patient-derived organoids on nearly all the tumors, and you can access them. As a researcher, you can set up requests. So this works. And clinician who is started in basic science, clinician want to use them to do predictive medicine, basically to predict what would be the best treatment for each classification of patients, as I told you, heterogeneity. Okay, but there's still things we can improve on this technology, and I will show you some examples afterwards. One big thing is that we don't reproduce the same thing in all the labs, because everyone has his own way to culture the cells, and it might lead to differences. So how do we establish standardized protocol? The other thing is as well, we need to do that, lots of cells. How can we use the minimum cell possible? So it's miniaturization, but also microfluidic, things that Morgan is developing. Another question we have is also, is the bank really representative of the concept? So I've talked to you about the heterogeneity, what we call the bulk heterogeneity. We take a tumor, we mix everything, we do an analysis. But now, in biology, we are, new trendy topic is what we call spatial biology. Okay, it means that in a tumor, one part will be different from the other parts. When we take a sample, maybe we take only this one, and this was a less aggressive part, let's say, and we don't take this one. Okay. So how do we better understand these different parts of the tumor, in particular for solid cancer? Yeah. And also next is how do we do that for all the patients? It's not done yet. Huh? What we do is we do it for representative patients. We classify the cancer and we say if the patient has this type of molecular alteration, then it's better to do that. Okay. And finally, what is not efficient yet for the clinics is the interaction with the microenvironment. It's still in basic research. We understand, we do co-culture of the tumor cell with the microenvironment, immune cells, let's say. We can, from the same patient, we also can take blood cells and freeze the PBMCs and derive immune cell from these PBMCs and do genic co-culture, but it's not routine, okay? Okay, when you are in a lab doing a research work, so you need to, you know all these approaches, so you need to take, look at what is best for your research question. So I focus on the patient's organoid, but maybe for the question you ask, using cell line in 3D is sufficient to answer your question. Okay. Because of the complexity of using organoids, you need to think whether it's really important to do that or not. And one thing which I didn't develop much is also in the issue of the matrix. So most of this is done in Matrigel, but we know we cannot standardize Matrigel, and there are lots of labs in the world which are developing standardized matrix to develop this algorithms. Okay. So the limiting points, as I see them, is high throughput, it's not working, standardization between labs, microenvironments, it's not working everywhere, everyone does his own way. One thing we are very interested in, Morgan and I, is reproduction of the mechanical environment of the tumor cell. So I'm not gonna explain much on that because it's gonna be the topic of the next presentation. One other thing I talked already is the spatial compartmentalization, meaning that a tumor is like an ecosystem. You have parts which are more have more oxygen, less oxygen, and they talk to each other. Okay. This current works, so we don't know how to do that. I mean, it works in mice, but we don't know how to do that yet. And that's what it's called organochip. Organochip, when we try to put in connection different organoids together. And finally, and this we are very far away from that, inter-organ modeling. Cancer is not a disease of the tumor. It's also a systemic disease. So the fact that you have a tumor changes your entire metabolism. You have cachexia, you have cachexia, you know what it is? Yes, the cachexia. Okay. You have systemic responses, you have circulating cells. We don't know why there's so much circulating cells, even at the beginning of the cancer. And this, we cannot mimic it yet in biofabrication. So one point I would like to highlight is the fact that when we do organoid, the type of culture medium you use will change your ability to predict the therapeutic responses. So this is a big issue. Okay, so 5 years ago, like organoids were going to solve everything, but now we're starting to see that it's not as good as what we think. Okay. So there's other things and so importance of standardization. And are we really mimicking everything in this type of protocol that I explained? And for example, in this article, they show that the gene expression in cell lines derived from patients put in 2D. Okay, so you take this tumor cell from the patient, you put them on plastic, you grow them, you make a cell line. Or you take the same cell, you put them in organoids. Or the same cell, you put them in what we call patient-derived xenograft. So it's a graft of the tumor cell directly in the mouse. Okay, and the patient, so the primary tumor. So what does better between these three techniques compared to the primary one? This is explained on this slide here. So this is you know this type of representation. Okay. So if this distance between, representing the distance in terms of similarity between all the samples. So in red, you have the patient-derived xenograft. Okay. In yellow, the organoids, and in blue, the cell in plastic. So you see, the cell in plastic look better than the organoids in terms of gene expression. It's a bit provocative what I'm telling you. It's just to explain you that organoids are not going to solve everything. And one of the reasons is that when you do organoids, to allow the cell to be maintained in 3D, you have very specific culture medium. So I just pasted you like the table to show you this complexity. So if you do an organoid from the digestive tract, from the appendix, the biliary tract, the colorectal, gastric, liver, so each time you don't have the same medium, okay? Same for breast, endometrium, okay? If you don't use this, it's not gonna divide. So what happens, and what we think is that this type of medium alters the differentiation level. And plus differentiation, change of differentiation is called plasticity. You've learned about that? And we know that plasticity is important for resistance to treatment. So here, by forcing the cell in a type of differentiation, we don't see the entire way the cells are represented in a tumor. Okay. Do you have a question on this part on organoids? Okay, second thing I wanted to talk about is about, so organoids is, you can do it with your hands, you don't need the machine. But then we are developing methods to handle these 3D structures, either using microfluidic apparatus, that's thing Morgan will talk about, or what we call bioimpression. So it's a different type of machines. You have 3 types of bioprinting machines, which use different technologies, where you have the cell in 3D in a liquid, and they are going to be embedded in a matrix. So the interest of doing that is that you can do spatial biology, put more cell at one part, less cell, and do mix cell types and everything. And in particular for bioprinting technologies, depending on what you want to do, I mean, the different techniques are interesting or not. I'm not going to go into details. We can read reviews on that. Okay. The rest of this bioprinting is that now, even in cancer, you can do co-culture and try to mimic, for example, have spheroids of tumor cells and have the cell endothelial cell make a tube and connect them. So you start to, okay, by bioimpression, you're gonna reproduce localization of the endothelium of the tumor cell and where the fibroblasts are. Okay. Okay, second part. Now, initiation of cancer. A bit more of background. Talk to you about pancreatic cancer, aggressive disease. But in reality, before cancer, there's different conditions in patients that increase the risk of developing this cancer. And this is in particular pancreatic inflammation, which is called pancreatitis. Emerging diseases which are called fatty pancreas. I'm sure Cathy Muller talked about adipocytes yesterday. Okay, we also have this accumulation of lipids in pancreas. We don't know really what it does. Breast cancer is well studied, but not in pancreas. And that's also what we call early steps, so pre-cancer lesions. And the issue here is we don't know whether they're going to evolve or not. So as I told you before, we need to better understand how this is important for developing cancer, to better predict which patients are at risk and try to diagnose it earlier. So when you work on this topic, here is a complex case. We have the initiating cells, which are called acina cells. They are the cells which are producing the digestive enzyme in the pancreas. These cells will have a first step, which is called acina to ductal metaplasia. They're going to change at differentiation. Okay. In this tissue, you don't have stem cells. So you cannot, like in colon, take only the stem cell and the genes, oncogenes. And then you have a mix of different early lesions that some of them can develop. So I've put you example, for example, here, and I'll show you some after as well in larger views to show you the complexity of all these steps. Organoid doesn't work in pancreas yet, but lots of people have tried. It doesn't work for neither for endocrine parts, which I'm not doing, diabetes, or exocrine. They have the same problem for the endocrine part. One of the reasons is that because there's no stem cell, we only have facultative progenitor, then we don't reproduce the heterogeneity of the organ. And if you do it with earlier step, you only get organoid of exocrine of embryonic pancreas. And embryonic pancreas, acina cell, for example, are not efficient yet. Bless you. Okay. So up to now, the only accepted model in papers when you want to study initiation of pancreatic cancer is the use of mice and genetically engineered mouse models. And that's what we have been developing, we have in the lab. But because we are doing lots of mouse work, I'm also developing alternative approaches. That's why I'm here today. And we try to, try other things to see whether we can reproduce better these initiation steps. Other techniques than doing organoids. So what are the genetic models that mimic this initiation step of pancreatic cancer? They are the following. They are called KC mice, KPC mice. Okay, these were developed by a researcher in the States, which is called David Clifton. I don't know if you know him, I don't know how many publications he has. He's very famous. His idea was very simple. He used a promoter of the gene specific of the progenitor of pancreas to express the main oncogene found in pancreatic cancer, which is a mutant KRAS. So KRAS, I don't know if you know this molecule, it's a GTPase that activates pathways, oncogenic pathways, PI3 kinase and MAP kinase pathways. When KRAS is expressed in the pancreas, you have pre-cancer lesion in all the mice at 6 months and cancer in only 10% of the mice. And when you combine them with mutation of P53, all of them get cancer. Okay. How we do that is we use a cassette which is terminating the transcription before the oncogene. And when the recombinase is expressed, this cassette is recombined and you have expression of the oncogenes or of the mutant P53. Okay. Just example to show you how it goes. This is the normal pancreas. Acina cells, the cells which are at the origin of the very aggressive cancer. Islet cell that control glycemia. If you do an inflammation, you get what we call the acina to ductal metaplasia, the first steps. but it's not sufficient to get cancer because it can reverse back. Okay. And then when you have mutated KRAS, that's what you see. This mouse, it seems to you that it has a lot of lesions, but it's perfectly fine. Okay. You can live with this with a tissue, and at my age, you have a precancer lesion in your pancreas. After 40, everyone gets precancer lesions. But not all of us have a cancer. Okay, so that's what we want to understand: why it developed, what makes it. So we use histology techniques to characterize them, and some of them look poor, like that, for example. Okay, that's what we want to reproduce by biofabrication. You see the complexity of what we want to do. That's the idea. Okay. And this is what you have in patients. Okay, you have the tumor, and around you have all these precancer lesions, and you have a little bit of normal pancreas that is left in the patient. And the patient, it was calculated that patients can live for 15 years with this type of early lesions, and we go undetected. Okay? And suddenly we burst. It's happened like that also in a mouse. You see nothing, and suddenly you see a tumor which is half a centimeter, and 2 weeks after, the mouse is we have to terminate the mouse. Okay, so it's like in patients. Okay, that's what we want to do. We fire biofabrication technique, because as you can see, for example, the main issue with the mouse model is weight, but it's very complex. So if we want to study small things, we're gonna have lots of mice to manage to conclude. We have to quantify everything. It's complicated. Okay. So it's long ethics, we can improve it. But it is powerful. If you demonstrate something with the mouse, it's gonna be square, okay? Still. So with this mouse model, what have we discovered? We have discovered, so I'm talking about the community, and we have participated into studying that, is that the acinar cell, I'll show you, initiate cancer through the mutation of KRAS, but also through inflammation. So you need inflammation to get this. And the first steps depends on the activation of PI3 kinase, which is PI3 kinase alpha. So that's my other dada. I don't know how to say that in English. I love PI3 kinases. So that's what we demonstrated earlier. And we also know now that if you have, so these steps is called acinar to delta metaplasia, it's cell plasticity, that sometimes it can reverse, but the fact that it reverses is not good either, you know. Your pancreas is look perfect, but the fact that you had pancreatitis before increase your risk of getting the cancer. All this was demonstrated with mice. And one thing we wanted to know is whether the remaining, okay, we know that only a few of these acinar cells do plasticity. We can see that in the mice. But what about the remaining cells? Okay, are they important? And why we thought that is because when we did this first paper showing that PI3 kinase alpha was important, okay, I'm gonna try to go through this figure with you. So you have amylase staining. Amylase is a marker of acinar cell, CK19 of duct cells. So acinar cells go into duct cells. T67 is a marker of proliferation. This is a normal pancreas, so you have lots of acinar cells and few duct cells. You do inflammation in context of mutation of KRAS or not, and you get acinar to delta metaplasia. That's suddenly the tissue become positive for CD19. You block this by inactivating PI3 kinase alpha. You see the cell try to express CK19 and do ducts, but they don't manage. They keep this acinar morphology, but they also proliferate more. Okay, so in the tissue, something happens. How do we study that? And when we look at this tissue in the context of inflammation, we know that both the acinar cell and the duct cell proliferate. Okay, so can we reproduce that in ex vivo? And we use bioprinting. Okay, my slide is a bit too complicated, but I'm gonna go through it. This is the figure to show you the cell lines we have been using. We're using a cell line, which is an acinar cell line that we can transdifferentiate using a cocktail of stimulus. You don't need to know what it is. If you're interested, I can tell you. And this is demonstration that we managed to transdifferentiate the cell. When we culture the cell in 2D, only the acinar cell proliferate and not the duct cell. So in vitro, in 2D, we don't manage to see what is in vivo. Okay. So we ask a question: if we put this cell in 3D using bioprinting, which is a way to standardize the way you are doing organoids, because you always put the same amount of cell, in the same amounts of matrix. You're very reproducible. What happens? Okay, we need to show that. So that's an example of the 3D growth of acinar cell in this bio paper. I always like they call that bio paper, but it's a gel. Okay. And when in this case, the acinar cell do not proliferate, and it is a duct cell that proliferates. Okay. So white is acinar cells, and purple, violet is proliferating. And what was really interesting is if we add a bit of acinar cell with the duct cells, they start to increase, to proliferate a bit more. Okay, so this to us mimics what we see in vivo. In vivo, we see the duct cells proliferating, surrounding by acinar cells. So that's what I wanted to illustrate, that biofabrication techniques are really good, but really you need to think, you really need to know your physiopathology to understand how it works, not to get too simplified model. Another thing I wanted to highlight, the next steps. I'm sure you've heard about the single cell technologies. You have a course on that or not? Single cell. So biologists, cancer biologists are nowadays. We love spatial biology and single cell approaches. That's our do it. And with these techniques, which arrived 6 years ago, nearly, that's how we discovered what we thought that cells were similar, they're not similar. So I'll show you slides with the pancreas before the acinar cell. If you look at them, they look all similar. But if you do gene expression at a single cell level, then you realize that they are not that similar. Roughly, they express differentiation marker, but there's differences. And that this discovery opened up lots of things, we thought about the change, new perspective on how we think the tissue is regulated. The hemostasis is regulated. So this is also true for pancreas. The complexity here is that acinar cell function is to digest nutrients, and in particular RNA. So doing RNA preparation of pancreas is very complicated, and at a single cell also. So this is just techniques, but still, this slows down the mitosis. So one other question we wanted to know, we see on the mouse that only a few acinar cells are doing, we express KRAS everywhere, only a few cells are doing metaplasia. Some of them are developing cancer. So the question is it due to this heterogeneity? And how the heterogeneity of this acinar cell is evolving? Okay. So to do that, we have developed a technique to do single cell preparation of the pancreas. I'm sure you have photos of that later on, no? I guess. And that's how the data shows up. Okay. So on this, what we call UMAP representation, each dot represents a cell. And depending on the genes which are expressed, we can say that they are mesenchymal cells, acinar cells, dot cells, etc. Okay. And we can, on this, you have color code that shows what I just explained. For example, markers of endothelium are specific to this population here, and I put here endothelium. Okay. And with these techniques, what we saw that the acinar cells are not homogeneous. Okay, that we suspected, that we demonstrated it. There's different what we call clusters of acinar cells. Okay. And I told you I love PI3 kinase. We use the PI3 kinase to modify the hemostasia of the pancreas and to understand how it works. So when we inactivate one of these PI3 kinase, you need to know what it does. This is genetics as well. What happens is that we change the distribution of the cell in these different clusters. Why I'm showing you this is in vivo. Okay, I'm showing you that is that when we do biofabrication, we use primary cells. We don't know in which state we take the cells. Okay, we need to. So how do we deal with that? And when we do our ex vivo culture, is it pertinent? What happens really in vivo? That's an open question. Okay. But what we see in vivo is that when we inactivate the gene, PI3 kinase class 3, which is called VPS34, we see a replacement of the tissue with adipocyte-like cells, as you can see here. Okay, the tissue is replaced. We have quantified it. Can we reproduce that in a biofabrication technique? The answer is yes. Otherwise, I would not show you. Maybe, or maybe I would not show it. I don't know. So we've used these techniques again, and we've done primary culture of acinar cell in 3D. And we managed to demonstrate that in this technique, we managed to culture the cell for 14 days, which is a technical challenge. And we mimic what we see in the mice. In the mice, we also see the lipidic accumulation at 2 weeks after the gene recombination. So there's hope for developing a better model of pathologies, even in precancer steps. The other thing that we are doing is that other question is in human, when cancer develops, not all the cells have KRAS mutation, only a few of them. Okay. So the question we want to understand is influence of the surrounding of this cancer-initiating cell in the fact that they will develop cancer. Okay. So to mimic that, we have tried to do using bioprinting methods to do lots of dots with a mix of cells with KRAS mutation wild type cell for the moment, and see whether we can do statistics on large scale to see what are the key factors for development of proliferation from the acinar cells. So this quantified here is just an example. We can see that in 14 days, the KRAS mutated cells, some of them are going to grow, and we quantify them just optically, showing that the dots are growing. And this allows us to develop high throughput analysis to test which are the factors which are important for KRAS development. So this is a work of the PhD who is in Bordeaux. And we have found out that it's the rigidity of the tissue is important to permits this progression. Okay. So I think I'm going to mention with this take-home message. I think technology is evolving a lot and very quickly. I've shown you like the first part, with the organoids, which are really now, if clinicians are using them, it means that it works really. Okay. But in basic research, you still need to improve the co-culture, how do we study relationship with immune cell. In pre-cancer research, I mean, we have tools, but we really need to develop specific things for that. It's not standardized. And just to explain you a bit the state when we started to work on this with the first paper I showed you, with the co-culture of acinar cell and dot cells. Then we say, okay, maybe we do a review on biofabrication techniques to study pre-cancer. And there was only 5 publications. Okay, so it shows you that it's not a developed topic, but I think it's quite important. If we want to try to reduce the amount of mice we are using, we really need to take this into consideration. Okay. Do you have questions for me?
No, it's more about a practical question.
Okay.
How many, do we have a lot of these printers, or are they just located? It's like highly special lab.
It's I think democratic. So for example, in Morgan's lab in LAS, there's all the technologies to do everything you want. In INSERM, I can tell you what is the strategy of INSERM. I started working on biointention because 10 years ago, INSERM decided to develop what is called the ART, technological units, on bioprinting, because it was the technology that was happening. And then they were going around France to see who would be interested, and I sent an application letter saying I want to do exocrine contrast. Okay, and my project was selected, so we started to do all that. So all the bioprinting that I show you is in collaboration with INSERM unit that only does bioprinting for INSERM labs. Okay, so you send a project and say, I want to study that, and they do the experiments for you. Okay. But their idea, so INSERM is funding that. But now what they want to do is say, okay, we have developed that, but now we need also to have to buy things in the labs. It's not yet, but I'm sure in 5 years, it's going to happen. At the beginning, it's complicated because it seems really easy to do what I show you. But it's 1 year of optimization because you want to know, you want your cell not to be viable. There's lots of things. And it's not something you can do without a really technical skills to understand what you need to do. And that's a big, so multidisciplinarity is great. I think that's what is going to solve lots of cases. But the big difficulty is that we get expertise from different people, and this is very good. Questions? Okay. Yes.
Bah, I retyped everything in English, but
Ah, tu veux que je te la...
ouais, so that is more English. But it's there's more and more technological platforms that are open for scientific research, but also for medical applications. In our lab, we have a very big technological platform, and it's not open only to researchers or only to CNRS, but it's open to insomnia because closed and in some in a way. And the prices are often very good when it comes to academia. So if you have any project in mind, I mean, there's a lot of open platforms like that. There's ours, but of course, there's not only our platform. And these sort of devices that you can print on or that they can be shipped also. So it's not a big deal if it's done in yes.
En fait, non, parce qu'il y a des personnes en ligne. Et le lien pour être en ligne, c'est ça le problème. For those who are online, we have a 2-minute break to put the presentation on the computer. I don't know, online you don't have.
Grazie the presentation.
It doesn't speak French, by the way. Who doesn't speak French? So it's better to take 5 minutes to have this conversation in English.
Yeah, we take 5 minutes to do it in English. C'est du roman. Hein?
Mais non. Si je l'ai ça va. Okay, let's start again. So okay, so in my case, I'm a physicist, and I'm working in a lab which is called the lab CRS. It's on the other side of the canal. And in our lab, we do a lot of biotechnology. And in our case, what we study is the impact of mechanical stress. And one specific tool that we use for that is microfluidics. So what I want to tell you about today is, first, I will briefly discuss the types of mechanical stress that can be found in the living, and just to be sure that we all have the same definitions. And there are some definitions I don't know if you are okay with, and I prefer that we all have the same definitions. That's very important. Then I will discuss microfluidics, and then I will discuss the applications that we have in the lab to this microfluidics. So if you have any questions, interrupt me whenever. That's totally fine. So let's start with the mechanics in the living. So there are three main types of stresses in the living, and a mechanical stress is always the same definition. It's always a force per unit surface. So force per unit surface is what often we call a pressure, and I'm pretty sure you heard about the pressure for atmospheric pressure and these other things. But globally in physics, it's always called a stress. So a compressive stress can be a pressure, but you have other type of stress. So one stress that we found very easily in biology is called shear stress, and that's the idea that you have a force that shears onto a surface. So it's a tangential stress. So it's as if you have something that flows on top of something else, if you want. So one of the main example of this shear stress in biology is, of course, blood flow. I'm sure you've all heard about blood flow. And because the size of the capillaries or even the veins is very small, and because blood is actually highly viscous, the motion of blood inside these small vessels will lead to a lot of forces that are experienced by the cells on that surface. So something to remember about shear stress, and that's different from the other types of stress, is that it's a dynamic stress. In order for a shear stress to emerge, you need motion. If you stop the blood flow, then the blood is static, okay, in the vein, and you have absolutely no stress that is exerted by the cells, experienced by the cells. Shear stresses can be found, of course, in a pathological condition, just like blood flowing, but you also have a lot of pathological consequences if the flow is bad. For instance, if you have atherosclerosis, when you have deposition of these, all of these lipids, then you have a reduction of the diameter of the vein, and that leads to aberrant flow. I'm sure you've all played with the hose of a water hose, and if you decrease the diameter of the hose, then the water flows much faster. So that's kind of the same principle. If you decrease the diameter of a vein, then the flow is conserved and the local velocity will increase, and that will increase a lot the shear stress. Okay. You can also have turbulent flow and all of that, and it's known that turbulent flow is actually super related to that type of disease. Another example that you may not have in mind is also every motion that we have in our body, which is related to torque. So every time I'm doing that, I'm going to have shear stress like that on the junctions, on the cartilage. It's also true in the knee, it's also true in the elbow, it's true in any motion that you can have in your body. And this shear stress is actually also very important, and actually also compressive stress, we'll talk about it right after. But this stress is also very important for the physiology of these articulations. If you don't have mechanical stress, then these articulations, they can start to rest, quote unquote. And you can have also bad depositions of matrix and sort of things that can lead to bad, how can I say, oiling of these articulations. So, okay, that's it for shear stress, and we don't need to talk hours about shear stress. The second type of mechanical stress is tensile. So shear is on the surface, it's tangential. Tensile stress is actually normal to the surface, like as we say in physics. So in that case, you actually pull on the surface. And it's still a force exerted on the unit surface. So you have a force that pulls onto a surface, and the intensity of that tensile stress will depend on the force and on the surface it pulls on. Okay. So where do we find tensile stress in the living? We also find it everywhere. One example, okay, so that's because I've retyped everything during this course, so there's going to be a lot of typos. I'm sorry for that in advance. So stress can emerge from the rigidity of the substrate. So I don't know if that's something that you are familiar with, the fact that cells on substrates of different rigidity, they have different behaviors. Is that something new to you or not? You heard about it? So cells that are on a stiffer matrix, most of the time they tend to be more spread, and they will develop more focal adhesion. So this is the focal adhesion schematically. And this focal adhesion, they are linked to the contractile actin cortex. Is that you all heard about that term, the actin cortex? That's very contractile. It's like an elastic that always wants to be pulled, and that's linked to these links there. So the higher the number of links, the higher the tension that you will have in the links. So if you plate cells on substrates that are stiffer, you will have a higher tension of the cell. And most of the mechanosensors that you have in cells, they actually respond to the tension. I'm pretty sure you all heard about piezo one as a mechanosensor. It was a Nobel Prize, I think, 3 years ago. YAP, all these sort of proteins, all of these, which are called mechanosensors, actually respond to the tension of the cell. And the tension can be modulated just by plating cells and substrates of different rigidity. So this kind of tensile stress, just like shear stress, where you can find at the cellular level, but also at the organ level, you can find at the cellular level. At the organ level, though, if you think of just breathing, every time you breathe, you increase the volume of the alveolar of your lungs. And the increase in volume, I mean, you don't create more cells when you increase the volume of your alveolar. So, of course, you stretch the cells. And when you stretch the cell, you will give them more tension. Okay. So you compress, stretch, every time you breathe. So this type of mechanical stress is also very important, and that's been shown actually in the living that if you inhibit this sort of motions, cancer cells can proliferate. And most of the time, when you have lung cancer appearing, it appears in areas of the lungs where the mechanical stress is impaired. So there's a tight coupling again between the physiology and the pathology there. One example that I'm pretty sure you haven't heard of in terms of tension is actually on the ear cell. And I kind of like that example because it's a very nice coupling between shear stress and tensile stress. So I don't know if you know how hearing works. No. So when we speak or when there's a sound, it creates a wave of pressure in the air, and that wave of pressure is basically, it has a frequency, which is the frequency of the tone of the sound, and that basically is something that oscillates. Okay. When that enters in the ears, that makes the I don't know that word, the tampon vibrates. Okay, the membrane at the bottom of the ear, it makes it vibrate and it oscillates at the same frequency of the sound. And these oscillations, it makes the fluid inside the ear into motion. So the fluid then oscillates also in synchrony with the sound of the wave that enters the ear. Okay, so you have a shear stress like that. You have you have a flow of medium inside the ear. And that flow of medium, it will create a motion at the surface of the cell because of shear stress. And the surface of all your ear cells, it has cilia. And these cilia, they will beat at the frequency of the sound through this coupling with the shear stress. Okay. And what's very, very interesting, I should use that for the people online. What's very interesting is that the cilia at the top of the ear cells, they have very tiny filaments like that are associated with membrane channels that will open when the tension is too high. So it oscillates, and when it oscillates in that direction, it's under tension that opens the channel. And by opening the channel, you create a calcium influx. And the calcium is then propagated into some sort of electrical signal. So that's how you do what we call in physics transduction. Transduction is the transformation of one signal of one form into another form. So here it's mechanical stress. It starts with the mechanical stress, and it ends up into an electrical stress, which is biologically encoded, of course, but it's an electrical signal, and that's transduced through this tension. So tensile stress, you can also find it at a molecular level. So it spans all the scales. Okay, and the last type of stress, just to be sure we all have the same definition, is compressive stress. So compressive stress is the exact opposite of tensile. So tensile stress you pull, compressive stress you pull. But again, you push on the surface, and that's a stress, it's the force per unit surface. Mechanically, it's very also intuitive, just have a surface and compress it. I have 3 slides, I think, just on a very, very careful note to explain the difference between this mechanical compression and what we call a hydrostatic compression, hydrostatic stress, if you will, and then osmotic stress. I don't know if you all know the differences. If you don't, then I hope you will know and remember the end of the talk. Otherwise, maybe you can then tell us. I'm pretty sure you've all been at the bottom of a pool or done that experiment where you basically just put your head under some sort of water. What happens when you do that? It compresses, the same word that I don't know, tampon. And you feel it. You feel the compression of the water. So the idea of hydrostatic pressure. So I'm explaining it with water, but that's exactly the same thing for air, just the air that surrounds us. The idea is that the deeper you go inside the water, the more you feel the weight of the water that's on top of you. So just imagine if you are under the water, there's a column of water that's on top of your head, okay? That column of water, it has a given weight, and that's going to put exerted pressure, again, onto a certain surface. And if you multiply this surface by the height of the water and the volumetric mass of the water, then it gives you the mass of water. It's the mass of this column of water. And of course, the deeper you go into the water, the higher the weight of this column of water, and then the higher the pressure you experience. So that's what we call a hydrostatic stress in physics. And that stress, you can find it also in the air. So if you go on the top of the mountain, you have less oxygen molecule and you have less pressure because you have less weight of that oxygen column. And the deeper you go, the higher the pressure. So deeper, higher, and higher in terms of altitude than the lower the pressure. So of course that exerted pressure, and you feel it, but what you feel is a mechanical pressure when you're there because it compresses the membrane. But the rest of your body doesn't really feel that type of pressure. So that's very different. It's not like you have something that's actively pressing on you. So the effect of hydrostatic pressure in the living is very, very different from the effect of mechanical pressure. What happens mainly is that you change the solvation of proteins and you play with signaling because you change the biochemistry of the proteins. Okay, you don't play with the membrane tension, for instance, of the cell. It's like we discussed for tensor stress. The second type of pressure, which is not a mechanical pressure, but is still pressure, is osmotic pressure. So that I think you're more familiar with in your studies. I hope you've heard about osmotic pressure. The idea of osmotic pressure is that it emerges when you have a difference in concentration between two compartments. So the classical example that we give is the one where you have a tube in the form of a U shape, and you have a semi-permeable membrane at the bottom of the tube, and it will let the water go through, but it will not let other solutes go through. And if you put on one side of the tube more solutes than on the right-hand side of the tube, then you will create a concentration gradient. There's not the same concentration on the left than on the right. And globally, nature hates when there's an imbalance of something, so it always wishes to balance things. And one way of balancing is to have a flow of water from right to left until you have the same concentration in the two compartments. But if you do that, then you have the difference in the height of the water, and that's a hydrostatic stress, a hydrostatic pressure. We just discussed it. So the weight of that column of water here is exerted on that, and that's what we call the osmotic stress. But it's of chemical origin this time, okay? It's not because of a difference in going deeper or again, mechanically pressurizing something. So osmotic stress. So osmotic stress, at the end of the day, it's going to be a water stress, but it comes from a chemical imbalance. And osmotic pressure is very, 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 very important because that's the basis of how all cells grow. Okay, the cells, it grows because it creates an imbalance between the concentration inside and the concentration outside. So to you, the cell concentration inside the cell is more concentrated than the outside or less concentrated? Who thinks more? Show of hand. One more, two more, and 20, I don't know. Or less concentrated? Less concentrated. So it's, more concentrated inside. It's more concentrated inside, and that leads to water going inside the cell. And that's how the cell can increase the volume. Okay? Actually, I'm pretty sure you've all heard about the tonicity of the blood and why it's very important to have salt also. That's because you have to maintain a good osmolarity. Red blood cells, these are very poor cells in a sense that they don't have any nucleus. They lost a lot of their regulation of what makes a cell something very alive. And in particular, they cannot regulate very easily their volume through osmoadaptation. That's the way we call it when there's an osmotic stress, we do osmoadaptation. So if the cell is under a higher osmotic environment, so more concentration outside than inside, then the water will leave the cell. So the cell will shrink. And on the other way around, if you have a smaller solute concentration outside and inside, then the water will go inside the cell and that can make the cell burst. So again, osmolarity is very important and that regulates cell volume. It can lead to disasters in terms of the blood, but it's also very related to how cells work. Okay, so mechanical compression is of very different origin. And again, as I said, it's just due to a mechanical stress applied directly to a surface. So again, if we come to articulation or for instance, the cartilage, when you walk just on the knee, here you have all the weight of your body that is applied on the knee, and that's a compressive stress that is applied. And that compressive stress, again, is very physiological. Actually, if you want to study chondrocytes, which are the cells that make this cartilage, it's been shown that their growth is much, much better when you compress the cells. Just because they are usually cultured, they're usually, found in under compressive environment, not under low stress environment. And that's something to keep in mind, and that's what Julie was saying. It's always about the local environment. When you study something, you have to study it and reproduce the local environment because it is their natural niche. Another example of compressive stress is the peristalsis during the digestion. So when you digest your gut, it creates a peristatic wave through a progressive compression of the cells, and that makes the bolus of food going down. But the cells in the gut, when that happens, they do feel that compressive stress. So they create it, they co-create it. The gut cells, that's the epithelium that does that. It's more the contractile fibroblasts that are surrounding and the muscle cells that are surrounding that. But they do feel also that compressive stress. So okay, for compression. One other type of compressive stress that is super found also in the living, and that's much less studied than the other types of compressive stress, is what we call growth-induced pressure. So the idea of that compressive stress is kind of simple. Anytime you think about cell proliferation, there's always cell growing. They grow and then they divide, it grows and divides, it grows and divides. But the cell proliferation never occurs in a free environment. There's always a boundary to that environment. And that's true for most of the cells. Only the planktonic cells in the ocean, they don't feel a limitation of their space. But otherwise, all the cells, they always have the limitation of their environment in terms of space. So what happens when the cells feel the border of the boundary of the environment? Well, either they just stop dividing, saying, okay, that's it. And that's been studied a lot in biology with the context of contact inhibition. Thank you for that maybe before. But contact inhibition is also related to cell density, but that's related to the fact that they feel some sort of mechanical stress when the density is high enough. That's how they feel that density is through mechanics. They can also in terms of chemistry, they can secrete a lot of stuff and measure the concentration through the chemical concentration. But actually, when it comes to cell proliferation, it's been shown that it's mainly the mechanics that matter than the chemistry. And if the cells wish to keep on proliferating, well, they have to keep. They have to push on themselves, they have to push onto the environment in order to increase the volume to keep on dividing. Because a cell will not divide if it does not increase the volume. Otherwise, the cell volume, the mean cell volume will decrease. And that cannot happen at some point. And so that's what we call growth-induced pressure. It's a pressure that emerges through growth. Most of our tissues in the body, or for animals, they are under equilibrium, meaning that they will not overgrow. But some tissues, they do overgrow, and that's the case of cancer. And when it comes to cancer, the cells, they proliferate, but they proliferate in a space where they don't have the space to proliferate initially. The tumor develops in the body of an organ. There's no space in that body of the organ. It was pulled and it was filled and filled without the cells. So what happens is that if they keep on proliferating, well, they will start to build up that mechanical stress. That mechanical stress, that growth-induced pressure, it has been measured experimentally. So we know it exists. And we know if we compare it to other organs, like the liver, for instance, that doesn't have any issue, it's stress-free, it's fully balanced, and there's no storage of that mechanical stress. But in the case of tumors, there's a high storage of that mechanical stress. We will discuss later the consequences of that mechanical stress. But one of the big consequences of this high compressive stress is that it can collapse the blood vessels inside the tumor. And most of the time, the tumors, they are polyperfused because of the mechanical stress that just clamps the blood vessels. And most of the work also in the clinic also aims at renormalizing that mechanical stress. Because if you have better tumor perfusion, then you can have also better therapeutic strategies. Just to show you one experiment that shows one of the effects of this compressive stress. So are you all familiar with the multicellular spheroids? Is that the first time you hear that term or not? Have you heard that before? Multicellular spheroid. It's basically a ball of cells. It's a very, very simple organoid. If you say it's an organoid to a lot of biology, they would say, no, it's not an organoid, it's a spheroid. And that's true. There's almost no complexity in that system, just a ball of cells. So it was super, super widely used in the 60s. It's still used for a lot of different researches. It's basically a mini tumor. It's in the size of the hundreds of micrometer, and that's, you can consider it as a tumor subunit, if you will. So these spheroids, if you let them grow, they grow, and if you put them inside a confining environment, just like agarose, for instance, so 1% agarose, that's a typical agarose that you can use for gels, and it's elastically confining this spheroid. And if you put it in, well, you see that the growth is super, super decreased. And that's something we will come back afterwards. The cells, they build up mechanical stress, they deform the agarose, so it's like they push on a spring. So if they push on a spring, they exert the force. You can measure that force, and that's the type of force that we can measure in time. So it's in the kilopascal range. That's the range of this force. And they grow, but they grow slower also. So that's something that we are investigating. And that type of mechanical stress, which is in the kilopascal range for animal cells, it can be found anywhere in the living. There's not only cancer cells that grow in a confined space. One of the examples that you have seen, I'm pretty sure, every day in your life is the roots of trees that can break concrete. So why do they break concrete? Because they grow in a space where they don't have the space to grow initially. And when the roots, when they grow under the earth, they need to push to displace the environment. And when they push, they generate forces. The forces generated by trees are much, much, much higher than the forces generated by tumors, luckily for us. And it's in the megapascal range, so 1,000 times higher than in the case of a tumor. And that's enough to break concrete. So just here, I wanted to show you the very first experiment that showed that tumors, they store that type of mechanical stress. And that one has been done in 2013 now, so a bit more than 10 years ago. And what the authors done is that they excised a tumor from a mouse, and then they punched inside the tumor, just like you would do for a biopsy. And what they have found is that the tissue relaxed in the center because it was compressed. Again, the cells that are elastic also. So you put more and more and more and more and more cells in the same place, so they compress themselves. And if you relax a bit, so if you give the cells some more space, then they can relax. And through this relaxation, you can measure the compressive stress. And if you do exactly the same experiment on the liver, well, you see absolutely no relaxation. So that organ, in that case, is not under mechanical stress. It doesn't have stored mechanical stress. But that type of mechanical stress we can find anywhere. So I told you we can find it in the roots of trees. We can also find it in some more, in some physiological cases during plant development. At the tip of the plant, what we call the meristem, there's cells that grow at the surface and they attach to the surface. And if they grow at the surface and they're attached, they will start to build up mechanical stress into it. And what has been shown is that there will be foci of building up of that mechanical stress, and that will lead to cell differentiation, and that will create the leaves of the plant. This type of mechanical stress, it has also a lot of physiological consequences. So there's always a crosstalk between the mechanical stress and also the cell signaling. We can also find it in microbes, in the case of bacteria and in the case of yeast, where yeast, they can also, I mean, all of these microorganisms, they can develop in biofilms. In biofilms, they are naturally confined. Biofilms are also confined in their environment. Biofilms growing outside, they will go inside the pores of rocks, or these kind of things, and they will build up mechanical stress. There's a lot of what we call biofouling, the deterioration in French, which is related to the fact that in microbes, they can generate a lot of forces. Microbes, they can generate as much forces as the roots of a plant, by the way, megapascal roots. A lot of forces. And these forces are relevant also for pathologies. In the case of that organism that we study with the Institut Pasteur, so Neisseria meningitidis, it's the name, it's the organism that's responsible for the meningitis. One of the questions is, how does it go through the blood-brain barrier? And what's known about that organism is that it's very sticky cells, and they can clog and occlude blood vessels. So if you have cells that grow and occlude the blood vessel, again, you disturb the blood flow. And if you disturb the blood flow, that creates clots, the cailloux. And if you create clots, that just basically closes the capillary where the cells grow, and then it creates a confined space. And the cells that will grow in that confined space. And we show that in cells there, they can build up a lot of forces and they can break blood vessels. And then supposedly that's how they would escape and go inside the brain. So again, that type of mechanical stress, compressive stress, that's relevant to cell physiology or pathology. Well, that's physiological to them. Just to finish on that part, in the specific case of this, we can find all the three types of mechanical stresses at once. So, as I said, there's a buildup of compressive stress. That buildup of compressive stress, it can clamp blood vessels, and by clamping blood vessels, it will create also a lot of differences in the interstitial pressure through the flow of medium outside of the blood vessels. And that creates shear stress. In the case of tumors, you also have often a change in the extracellular matrix. It's a big inflammation, so you have a lot of fibrosis that leads to matrix that is often much, much stiffer. And as I said at the very beginning, if cells are cultured on a stiffer environment, they experience locally higher tension. So you also have tensile stress in the case of tumors. And in terms of compression, so you have the growth pressure, but you also have one type of pressure that comes directly from the matrix. In the matrix, and especially in the case of pancreatic cancer, you have a high deposition of hyaluronic acid, which is a type of component of the extracellular matrix. And hyaluronic acid is negatively charged. The way it works usually is that water brings counter ions to make it neutral. But if you have problem with the water flow and high deposition of that negatively charged chemicals, when that creates electrorepulsion, the negative plus in front of the negative, they push. And in terms of chemistry, we call that electroswelling. So the matrix swells. And that creates compressive stress because the matrix swells, but again, in a fixed volume. It cannot increase the volume. Cool. Maybe I can just do that. Okay. So just to conclude on that part of mechanical stress, okay, pretty sure that was a problem. So first shear stress. So shear stress, it happens to the motion. You always need to have motion to have shear stress. It's a dynamic stress. It's always onto the surface. And for instance, you have water flow, blood flow, and everything that's related to fluid flowing will lead to shear stress. In the case of tensile stress, it can emerge through the stretching of cells, but also through adhesion, for instance, of stiff substrate. Compressive stress. So again, there are three types of compressive stress. There's hydrostatic, there's osmotic, and there's mechanical. And they are very, very different in their origin, and they have very, very different consequences also on cells. And they are integrated in terms of signaling very, very differently. So if people say, if you hear a researcher talking about pressure, just make sure you understand the type of pressure that that person is talking about. For instance, I'm working on mechanical pressure, and I tend always to say pressure, but that happened to me that someone in the audience had a confusion. I thought I was talking about hydrostatic pressure. So for 45 minutes, there was a big miscomprehension just because we were not talking about the same type of pressure. So now I always define what I'm talking about, but also keep in mind that there are different types of pressure: hydrostatic, osmotic, and mechanical pressure. In the living, for animal cells, every type of mechanical stress is always in the kilopascal range. And just remember that term. So shear stress, tensile stress, compressive stress, the rigidity, all of that, it's always in the kilopascal range. For plant cells and world organisms, basically, it's going to be hundreds of kilopascal or megapascal, the physiological range of pressure. But in case of animal cells, it's the kilopascal. Of course, you have examples both in the case of physiology and pathology. So, was that part clear? Or do you have specific questions? Cool. So now to something that you may be less familiar with. How do we reproduce this type of mechanical stress in the lab? So Julie told you a lot about biofabrication, bioprinting. What I will be talking now is about macrophysics. Know that there's not one that is much better than the other one. It's just different techniques that you can use depending on the applications that you want. What is true is that anyways, classical cell culture, it doesn't have dynamic control. We know that the chemical and the mechanism can change in time. It's steady conditions. It's always in 2D. It's on a very steep environment. Anyways, it's not relevant for cell culture. And most of the consequences, most of the conclusions, that you can get from just culturing cells in 2D are going to be most likely wrong when you want to compare them and to conform them to the living. In some cases, it's very interesting. But in most of the cases, it's going to be wrong depending on what you want to do. So if you approach physiology or pathology, it's going to be wrong. So how do we control the chemical environment and the mechanical environment? So in the lab, we use a lot macrophysics. So macrophysics, the definition is very easy. It's just the study and the control of the fluids and the motion of these fluids at the micrometer scale. The micrometer scale is the scale relevant for most of the fluids in the living. You have examples in the case of animals, so the veins, the capillaries, all of that. It flows in micrometer scale vessels. But you have also other examples. For instance, the veins of the leaves is the same size, or also the silk coming from the worm. It's also in that case in the size of the micrometer. So one slide history of macrophysics. So there's no point maybe of remembering it. I just want to explain how it emerged and just basically briefly saying that it emerged from the combination of two fields at the same time. On the one hand, most of the chemists, they were starting to work on chromatography, in particular gas chromatography. So they were starting to control the flow through tubings, because they need to control the flow through tubings, having very complicated circuits of fluid, water or gas, fluid, and creating valves and all in order to have all of these fluids going at the right place at the right time. At the same time, there was the emergence of the macro technologies and in particular MEMS, but everything that was related to what we call the Silicon Valley. So why Silicon Valley? Because most of the macro technology, they use silicon. And one thing that was highly developed in the 90s is what we call MEMS, for macro electromechanical system. And these systems, these are basically systems that make a motion when you have an electrical input. For instance, the watches, that's how they work. You give them electrical power and they make a mechanical motion. So it's a MEMS. And these MEMS, we create them in a clean room and in very, very clean environment. We call that a clean room because it's a room where there's absolutely no dust. You have most of the dust that are much bigger than the size of what you study. So it doesn't work if you have a lot of dust. So basically the idea and what happened in the end of the 20th century is to combine these two approaches. The idea was through these macro technologies, develop molds on the micrometer scale to create channels, and at the same time use all the control of the fluids that you can get in order to have the flow going through these macro channels that you then create at will and not just through tubing. And that's how macrofluidics emerged. And it emerged with the promise of reducing a lot of the volume, and there was a lot of ideas about a lab on a chip to reduce all of the things that you can do in the lab in a tiny chip in order to be faster and cheaper. Doesn't work. But that was the goal at the time. So how can we do a macrofluidic chip? So as I said, the idea was to use everything that we could do in macro technology. And what we can very nicely and easily do in macro technology is a silicon wafer. So a wafer, a galette en français, is something that has about that size. It's in silica. And what you do to create this sort of wafers is that you will add on top of the wafer photosensitive resist that has a given thickness, and then you will use light to pattern that resist. And it's exactly like photography. The idea is that only the part that is exposed to the light will change its properties, and the part that is not exposed by the light will remain what it was before. So just like photography, basically through light, you will pattern a given surface, but here that surface is in 3D, it's a bulk. And then if you develop, you will wash everything that was not exposed, and you end up with lines. Here it's just one line, but you can do whatever and more complex structures if you want. Then when you have that, what you can do is pour a material onto it. And the material that is the most widely used in macrofluidics is called PDMS, or polydimethyl siloxane. It's not important to remember it. It's a silicone. So silicone is biocompatible. You can find it anywhere. There's in shampoo. You have some in the breast implants. It's the PDMS. You have them in Coca-Cola. So you even drink PDMS. Just to tell you how not dangerous it is. It's not the sugar that's dangerous. Okay, Coca, don't drink Coca-Cola. But if you drink Coca-Cola, there's a bit of PDMS. It's very nice for emulsion. Anyways, so that PDMS, it can be solidified. And when you mold it and you take it out, then you have basically replicated your lane in volume. Then what you can do is that you can punch holes in that, you can bound it to a glass slide, and then just like that, you have created a macro channel. And you can have fluid going through that macro channel. That's very simple macrofluidic chip, but that's one of the types of macrofluidic chip that is called the first one. And then if you put liquid in them, well, you can have very clever geometries and all, and you basically flow through. Okay, so that's the basics of macrofluidics. So what can we do with it? One of the first things that were done with macrofluidics is chemical control. Because of course you have pumps, you have a chemical that you flow inside the channels, so you can decide to what to flow, when to flow, and how to flow it. One of the nice examples that was done, for instance, is to have a temporal control of what you flow. And here they made oscillations of, for instance, glucose and galactose, and to show how cell would react to that in terms of signaling dynamics. But most, in terms of what has been done more recently with this sort of devices, basically what you can do is also you can temporarily pattern the substrate. Just like during development, you have temporal waves of wind signaling, for instance, that will come and go. You can also have wind flowing in and flowing out, and you can see how that can affect cell differentiation in that system. Because you can temporarily control the chemicals that you flow in. Very interestingly, you can also spatially control the chemicals that you flow in. You can have very clever geometries using 3 channels like that, in which in the channel on the left, you can have a green flow 4. In the one on the right, you can have a red one. In the channel in the middle, you have absolutely no flow, but it's just connected to the side through very narrow channels. And when you do that, you're going to have through diffusion, the red going to the left and the green going to the right. And you will create a concentration gradient like that, and a steady concentration gradient. And here you have the concentration of the green, the concentration of the red, and you see that they mix, and at the center you have the equal concentration of the other two. That also has been widely used to study development when you have a lot of gradient of morphogenes during embryogenesis. And this sort of gradients can be reproduced a lot in macrophages. So it's again a very convenient thing. I also wrote here, so post chemotaxis, you can also check if the molecule of the cell, sorry, will move more to a given concentration of a given molecule. You can also use it to do drug testing. If you want to see at which concentration the cells die, when you receive the cells are everywhere at the beginning, and I don't know, they all of these are dead and these ones are not dead, and then you will know that okay, at that concentration the cells die. That's also used for this sort of assay. Of course, something I think intuitive, because you are in a channel, you can control the shear stress, but that's how fast you flow the culture medium. So one of the applications that you can have is you can just put endothelial cells. You can have a shear stress like that, and you can see what happens to the cells. Okay, so something maybe less intuitive is that the PDMS that we use, this material, is highly deformable. So because it's deformable, it can be stretched in particular. So some devices that are not directly macrophilics, but related to macrofabrication that has been developed is what we call cell stretcher. So you can place cells on a substrate, on very PDMS membrane that is highly deformable. You just pull on the membrane, and by pulling onto the membrane, you will stretch the cells at the surface. There's been a lot of applications there to see how cells reorganize, to see how they can create stress fibers. You can also do it on a dense epithelium. So you culture cells until they fully confluent. And what the researchers have done is that then they stretch, so you free space, and then you see that the cells re-enter the cell cycle very, very fastly. You can also do the converse, you stretch and then you relax, and you see that all the cells, they massively die when that happens. So that allows you to study the impact of tensile stress on the cell. One thing that I want to talk about, I'm not going to talk about it a lot in the presentation, but I think it's very important to mention is what we call organs on chip. I don't know if you've ever heard that term. So you heard about organoid, okay? Have you heard about organ on chip? Yes? Or only one yes? So organ on chip, so organoids, 10 organ on chips that were developed in the 2010. The two big names are Hans Clever for the organoids, was the one who developed the first organoid from the gut, if I remember correctly. And the big name in terms of organ on chip is Donald Guillermet-Guibert in Harvard, and he developed what we call the first organ on chip. So the idea of organoids and organ on chip are the same, in the sense that for organoid, you add the chemical environment in a matrix. That's nitrogen most of the time, but you don't enforce the geometry. You don't add any specific mechanical stress. You just hope that by adding the good chemical at a good time, the cells will self-organize and go to the good function. That's an organoid. Organs on chip, they were developed more by engineers than by biologists, and the idea was exactly the same, but instead of playing with the chemical, you play with the mechanics. So they were thinking that if you give the cells the relevant topology, for instance, for gut, you give the crypts and the villi. For a lung, you put cells in a stretching device, etc. Then the cells will self-organize and also go closer to the function that they were supposed to have at the beginning. So again, the idea of self-organization, but through mechanical control in that case. That's what we call an organ on chip. Nowadays, the two fields, they start to converge because organoids, they have a lot of limits, in particular because the topology and the mechanical stress is not necessarily reproduced. And organ on chip, they also have the big limit that the chemical environment is often super not relevant. I just use the classical DME. So the idea is to take advantage of the two fields at the same time. So then to come back to that particular example, Donald Guillermet-Guibert, what he developed as a macrophilic chip, because that's a macrophilic chip, is a lung on a chip. So the idea of that chip is that it has a suspended membrane that can be stretched like that, and that's in between two big channels in which you can have a vacuum. So if you have vacuum, then the channel volume will decrease, and that will stretch the membrane. And you can do cyclic stretch. Here it's a real-time movie. But what he did, which is also very, very interesting, is because you can control the flow, he put at the top lung cells, and they're connected to air, because lung cells, they are in the air. And at the bottom, he put epithelial endothelial cells, so that are connected to culture medium. And he saw the interaction between the cytotype cells. And this is through this sort of device, which I've discussed at the beginning of the course, that I showed that, for instance, if you mix normal lung cells with cancer lung cells, if you put absolutely no stretch, then the cancer cells, they will overtake the population extremely fast. But if you put stretching, then the cancer cells, actually, they are extruded, and then they just die. Telling you that there's cancer cells, and that's what Julie was saying, we have a lot of precancerous lesions, but naturally also the body has a lot of ways to dealing with these lesions. And maybe that's also one way through mechanical stress in that case, for these lungs. In terms of compression, well, the idea is kind of very simple also, is that we can put in a channel like that, cells, and on top of the cell, you just put something that can be deformed to just squeeze the cell, like that. So that's being used a lot in mammalian cells, a lot in yeast also. And here you see this, so here you have the pillar, which compresses the cell. It's in this cell, and you see that if you compress the cell, you see the area increases and the height decreases because the volume is conserved. Okay. So in terms of macrophysics, it was super quick. The idea was just to present roughly the type of technology that it is and the type of things that we can do. In terms of fabrication in a clean room, what we often say to students who start to do macrophysics is that you are mainly limited by your imagination. Now we can do a lot of very complicated stuff. We can do macrophysics in 3D thanks to bioprinting as well. We can do a lot of things about the formable that we can incorporate. We can also have a macrofluidic chip into which you can add a hydrogel and polymerize the hydrogel inside through photopolymerization. You can add cells whenever you want. You can do so many things. The idea is that always think of what you want to do and how you want to reproduce it, but most of the time there's always a technological solution to do it. So we do all of that in a clean room. So to answer half of your question earlier, creating this sort of tools, it requires something that is extremely clean. So a bioprinter, everything is printed inside the bioprinter. And these devices, they tend to be cheaper and cheaper, in a couple €100,000, which compared to the equipment that we have in our clean room, I can assure you is very cheap. But if you want to do something that is a bit more complicated, that needs the device to be taken out, to have extra steps, all of that, to add a sensor inside it, then you need to do it in a very clean environment. To avoid the physical contamination with dust, for instance, but also to avoid actual biological contamination, because this device, then it will be used, and most of the time these devices, you cannot autoclave them, you just destroy them if you autoclave them. So you also need to create these environments in a very sterile environment. And in a clean room, there's no dust, but there's no bacteria. It's so clean that actually during COVID, it was a very safe place to hide. Of course, the lab was closed, but there was no reason. And we have a lot of different materials that we can use. PDMS is a great example because it's highly deformable also, and you can, and it's very cheap, and also you can do a lot of shapes with it. And if you want to learn more on something that I haven't really explained, which is how to make a mold, you can always go to a Sylvain video on YouTube, where he did, so that was for his master's course. He had to do a video of how to do something, and he just went to the clean room, and it's a 5-minute video on how to do it. It's kind of funny and interesting at the same time. Okay, so before I move to the last part, which is kind of the applications of what we do, but I can go much quicker on that. And do you have questions on that second part on the macrophysics? What can be done, what cannot be done? Okay. Okay. So in the rest of the presentation, there's 10 slides, it's going to be 10 minutes maximum. I want to talk about what we do mainly in the lab, also in collaboration with Julie, which deals with this growth-induced pressure and how it impacts cancer cells. And just to tell you that in the lab, we create these macrophilic devices to compress cells. So the idea to study growth-induced pressure is to create a cage because you want to confine the cell. So the type of cages that we develop in macrophilics is basically a cage that is connected to a lot of narrow channels on the side, and these channels basically are such that the cells cannot go through because you want to confine the cells, but the chemicals can go in, so that you have a fixed chemical environment. We don't want to mix mechanical and chemical stresses. We just want to have one type of stress, which is mechanical. So we put the cells in, then we confine, we close basically some sort of a valve, and the cells, they grow, they build up mechanical stress, and we have force sensors to measure that mechanical stress. And at the same time, we can image everything. So we built, and I will go fast with that, we built a device like that in the lab, and here you have an image of that device. So it's very small, and you have the channels there, and you have different elements that you can insert. And the idea of that device is that I know why it bugged because of that video. Okay. And the idea of that device is that we can flow a spheroid in. At the beginning, it's a sphere. Then it enters the chamber, which is confined. Then it morphs into some sort of cuboid, becomes cubic. And here you see all the tiny channels. So it's well fed. And actually, when we look at the proliferation of in the sphere shape, it's exactly the same as if you were to culture it outside of the chip. And then it grows and grows, and it pushes against a force sensor. And we can measure the force in time. And here you can have the pressure, as a function of time, of that spheroid that is confined. And basically, the pressure increases. It's in the kilopascal range, similar as what I told you before. And here we have a very, very high resolution imaging if we want to combine that with different types of measurements that we can do in the lab. So these sort of devices, they are not very easy to build, but they are an example of what we can do from only instance. So one thing that I presented at the very beginning in the effect of this mechanical compression, and I think I'm just going to say compression, but okay, I'm talking about mechanical stress here, mechanical compression, is the fact that the cell proliferation decreases. And that's something that we find in any organisms that we study. In the lab, we study bacteria, we study yeast, and we study mammalian cells. And in all of these three organisms, every time we confine them and they build up mechanical stress, every time the growth decreases. And that's an obsession to me, which is why that happens. And I'm going to explain to you why that is important to understand that response. So if we take spheroids again, really it can grow, and if it's embedded in agarose, it grows, but much less. Something that we have started to look at with Julie is, I forgot to translate that slide, sorry. So it's only in French, but I'm going to guide you through it. Well, I think it's pretty explanatory, but still. Is the effect of classical chemotherapeutics that I use in the case of pancreatic cancer. And one of the one that is extremely used is gemcitabine. So gemcitabine is a CTP analog. It's a deoxy-CTP when it enters the cell. And basically, during the synthesis of DNA, it can be incorporated in the DNA, and that creates an irreversible error that leads to cell apoptosis. So the cells need to proliferate and synthesize DNA to be sensitive to that drug. And anyways, most of the chemotherapeutics that are used are basically targeting cells that do proliferate. So what we wanted to see was the effect of that classical drug when the cells are compressed, because pancreatic cancer is a highly compressed cancer. So first what we did is to add that drug to spheroids. And here you have the normal growth, and here you have the effect of the drug. But basically, when we add the drug, it kills the spheroid, which is what you would expect. Here it's 12 days. If you were to wait until 30, 40 days, but it's very hard, so we don't have a lot of statistics because of contaminations to maintain the cells and image them, so they cannot incubate, blah, 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 for so long. A lot of bad things can happen. But basically, what happens is that at some point, you have no cells, everything is dead. Okay, now what happens when you add compressive stress? So when you add compressive stress, here it's a free spheroid subjected to gemcitabine, and here it's a compressed spheroid subjected to gemcitabine. You can just see by eye that the drug almost doesn't work. And here you see the volume of these spheroids as a function of time, and it almost doesn't change. It remains roughly constant. So the good thing is that it doesn't proliferate more still, but it doesn't do what it's supposed to do, which is killing the spheroid. And the answer is why. And so I'm a physicist, and we do also a lot of mathematical modeling. And what we decided to try first, because if you want to investigate that why, you can say, okay, I'm going to see if this pathway is turned on and then blah, 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 but there's so many pathways that, okay, where do you start? So something else that we decided was, okay, can that be predicted if we say that compressive stress decreases cell growth, and here you have the volume of the spheroid increasing in time. Under compression, it's much less. And knowing the effect of the drug in the free spheroid, which is the yellow curve here, how much it's supposed to kill a free spheroid. And then if you say that only the cells that are proliferating are sensitive to the drug, you can say that because the cells proliferate less, maybe the drug will kill less because you have less target cells. So we can mathematically model that, and we can predict, because we have the numbers, what would be the combined effect of the two. If we assume it's purely linear and there's no signaling involved. And here it's the magenta dashed line that you see here. So here it's a prediction. And the prediction is that it's supposed indeed to kill much less. And that prediction, and that's also one message I wanted to give you, is that when you do multidisciplinary research, all of the different fields are very interesting. That field, for instance, here of modeling and physics, it allows to avoid you studying thousands of pathways that actually would not be relevant to that. The only pathways that are relevant to understand what happens here are the ones that decrease cell proliferation, because it's because you decrease cell proliferation that happens. And that we have checked with other drugs, and it works exactly the same thing. The same way we also check with other type of mechanical stress. Basically, any type of mechanical stress that decreases cell proliferation will lead to a lower efficacy of the drug. But that makes sense, right? Because all the drugs target cell proliferation at some point. So the big question is, can we overcome these effects? And to overcome these effects, we need to understand why proliferation decreases. So that's what we do in the lab. And as I said, this type of mechanical stress is experienced anywhere in the living. So in particular, we studied in the yeast Saccharomyces cerevisiae. That yeast is found everywhere, and just like PBMS, you have eaten or drunk a bit of that yeast in your life. There's also wine if you want. And there's a lot of similarities in how the cell divides and how the G1S transition is regulated. So I guess you're all familiar with the cell cycle and G1S transition of the cell cycle. One of the things that we have found is that when the cells are compressed, they are always stopped at the G1S transition. So the question is, why G1S transition? And the yeast G1S is very, very similar in terms of the proteins than animal cells. So yeast are a very nice model system to study this sort of question. And we also build up mechanical stress for that, devices, macrophage devices to study that. And the cells build up mechanical stress in these devices, and you can also study the response of the yeast under mechanical stress. So to conclude, because I don't have any conclusion on why cells grow slower, we have a lot of hints, and I'd be happy to discuss it. Mechanical stress has a lot of consequences in the living, and it's ubiquitous. You can find it everywhere. It has a lot of consequences in terms of physiology, but also in terms of the pathology. Here you have another yeast, which is called Candida albicans. It's a gut commensal. And that commensal can become a pathogen. It's called a commensal to pathogen transition in the gut. We don't really know why, but we know that it's highly confined also in the gut. And at some point, we have, so that's from our lab, we see that yeast can morph into this highly invasive and pathological filaments due just to mechanical stress. So mechanical stress is also implicated there. And to study mechanical stress, you truly need some pluridisciplinary approaches. There's a wide array. You need engineering approaches to build the tools that you want to build and these tools are very different depending on the question that you have. And depending on the cells, depending on the organ, depending on everything, you need to build a specific tool. And this tool can be built through bioprinting, through macrophysics, all of that, but it needs to be specific at some point. Of course, you need the biology. I should even put it before because that's where the question comes from. Why does something behave like that? And these biology approaches combined to the rest allow you to understand these fundamental principles that you can tackle by playing with the mechanical stress or the chemical stress. I think physical approaches are also very important because they allow you to build models on what you see. And these models are also experiments because then you can test the model. You can say, okay, if I have understood well, then this should behave exactly like that. And that's what we have done with this chemotherapeutics. And of course, at some point, the goal is to go towards medicine. And what we want to do also with Julie is to translate what we know to the medical world. And in particular, can we use the knowledge that we have in terms of the compressive stress, in terms of diagnosis, or in terms of therapeutics? After all, compressive stress, it decreases cell proliferation. Can it be good to treat cancer? Does the fate of a tumor depend on how much it's compressed? That would be important in terms of diagnosis. So that's also the type of question that we try to bring to the clinic. And macrophysics is good for that. Of course, but help on macrophysics. And to conclude, that's my very last slide. If it goes. I just want to finish on that. And this was built in the 19th century, so a very long time ago. And this device is called a compressive apparatus. And this device, it was built by this French clinician, Guillermet-Guibert. And that was at a time where there was no surgery, there was no chemotherapy, there was no radiotherapy, there was no therapy basically for cancer. And his idea is that, but people knew about cancer, of course, they knew about tumors. Tumors, it comes from the Latin word growth, and even the Roman, they knew about tumors. Basically a growth, so you can feel that growth. And the idea is that you see a growth, you just want to, okay, press on it. So the idea of that device was to say, okay, what happens if we compress a tumor? But not compress as in leading to a compression that will be above the systolic pressure. The idea is not to do, to create some necrosis of the tissue. The idea was just to compress a bit. And what he published in that very interesting book is the result of his experiment on breast cancer, where he found that in 50% of the cases, the growth that he measured, and I won't say growth because there was no characterization, so we don't know exactly what that was, but it was still a growth. It decreased or totally disappeared when he was doing that type of experiments. In a good 50%, there was no change, no progress, no regression. And in 1% of the cases, actually, the patient died faster. Maybe it's related to the fact that we know that compressive stress can also trigger ENT. But now we know this other things, before they didn't. So all of these studies, they fell into oblivion because surgery happened and they decided to cut the breast faster. It didn't work, you know the story. But his ideas of using mechanical stress as a therapy was there also a long time ago. And in a way, why not also use that again in the future? All right, thanks. You have questions now on this last part.
Really can you talk about also can you device and like
so you can have there's a flow. You can take the flow that was through the sample and you can analyze it. Now what's happening also is to embed directly this device. You can have optical filters or maybe
Feedback on our presentation.
And we have the lights.
Things you want them to do for the next month. So they will have this Yeah, we will put it. It's okay. Thank you then. Bueno, on n'est pas du tout des intervenants. Oui, c'est bon. Qui est responsable de ça? Ce qui s'est passé, c'est ça.
Il avait Camille, je sais pas si vous l'avez croisé. Voilà, dans ces places. Non.